Okay, shall we start? Allison, you ready? Okay. So, hello everyone. I do recognize most of you. I think all of you. Nice to see you. I'm very happy that we have Dr. Allison Bradbury today um, to talk about to us about what it takes to get to develop treatments and to get to where we all want to go to clinical trials and approved therapies. And as I said, what it takes, what we have to do before we can do that. Uh, I, Alison will talk about her role at the university, which is something she can do better than I am. But I want to say she has been involved with patient advocacy with many groups in neurological disease. So it is really great to have you here. And I hand it over to you. Thank you.
Yeah, thank you, Valerie and Keely, for inviting me to join the Yaya Foundation today. As Valerie mentioned, my primary hats I wear during the day is that I'm a principal investigator in the Center for Gene Therapy at Nationwide Children's Hospital, and I hold dual faculty as an associate professor in the Department of Pediatrics at The Ohio State University. And then a second role that I have is that I sit on the board of directors for the American Society of Gene and Cell Therapy, or ASGCT. My lab is highly translational in nature. So, what that means is that we work to understand inherited pediatric neurologic diseases in order to try to develop genetic therapies that we eventually translate to the clinic. As Valerie mentioned, I've been very involved in patient advocacy. So, I just wanted to highlight some of those roles briefly before we dive into the science. So I did my graduate school work, my PhD at Auburn University, where we focused on two inherited pediatric neurologic diseases called GM1 and GM2 gangliosidosis. And we developed what's called an AAV-mediated gene replacement therapy that was translated to the clinic for both of those indications. And I now serve on the scientific advisory committee for the National Tay-Sachs and Allied Disease Association, which is how I have met Valerie. I then went on to want to work on another disease that is similar to GM1 and GM2, but down the same lipid pathway, and it's called Krabbe disease. So I worked on the development of a gene therapy for Krabbe disease, which we also translated to the clinic. And I also sit on the scientific advisory committee for Krabbe Connect. And now I have my independent lab at Nationwide Children's Hospital. And one of the indications in my lab that I'm working on is another leukodystrophy called vanishing white matter disease, where we're still in the preclinical stages of that development. And that's how I met Keely through the United Leukodystrophy Foundation. So that's some of my kind of role with patient advocacy and patient foundations. And I will just like shout out to all of you that you really help drive research, the knowledge that you bring as parents, and further educate research and drive research is always really motivating and inspiring. And a lot of the work we do couldn't be done without the support of various foundations. Okay, so I'm going to take us all the way kind of back to the very beginning to really help kind of level set and preface my talk about the development of genetic therapies. And we really have to start with what is called the central dogma of biology. And this is really just the principle that our genetic material, DNA, is transcribed into RNA, and RNA is then translated into a string of amino acids that you might depict like this like bead on a string. But then really those amino acids fold into a really complex functioning unit, the protein. The way that RNA becomes a protein is in these 3-letter codes. So you have 3 letters of your DNA and then RNA, and based on what those 3 letters are determines what the next amino acid is, or which is the next bead on the string, if you will. So for example, if you have an A, and then a G, that indicates for the bead to be a lysine. So that is the code of our genetic material that determines what the protein is. And this code is very important, obviously. We have specific codes that then stop and say, this is the end of our code. And now you have completed the important instructions for how to make the protein. However, as we know, we can have these misspellings, these changes in our genetic material that can impact the structure or function of our protein, and this can result in disease. So, for example, you can miss a whole codon, a whole 3 letters, and that would mean you're missing a whole bead on a string, and that could result in a shift of how the amino acid is made. You could have an extra letter, and again, that would change the order in which the 3 letters are read, and that can kind of mess up the instructions, how things are spelled and read. You could have one missing letter or just one misspelled letter. So there's many different ways that you can have these changes to your DNA that result in an altered protein. Some of these changes mean that your protein is just chewed up, it's degraded. Other changes might mean that your protein is shortened. And some of these changes might mean that your protein takes on a different structure or folds different or a different function. So we have these changes at the genetic level that impact either the structure or function of our protein. And this is what results in disease. So now that we know that we have these molecular changes, really the question is, can we now fix this genetic defect? And if we can, what's the best tool? So if you have a molecular change, a misspelling in your DNA that results in what's called loss of function of your protein, that means your protein is absent, shorter, or completely degraded, then you might need a therapeutic strategy that's adding in a new, healthy, functioning copy of that gene to make up for the deficient protein. You could also, particularly if your mutation is in this junction called a splice site, which is where your introns and your exons meet. Introns are part of your DNA that are usually cut out by little molecular scissors. It's an important little junction there. If your mutation, for example, is at a splice site defect, these could be amendable to something called an antisense oligonucleotide, which acts like a little band-aid to cover up that misspelling. However, say that your molecular change, your variant in your DNA results in what's called a gain of function. Instead of too little protein, you actually make too much protein, which results in toxicity. In that case, you don't want to make more of it. You don't want to give more of the protein. You want to actually knock it down. So in those cases, when you have a gain of function mutation, your therapeutic strategy is going to be very different. You want to do gene knockdown so that you reduce the amount of that toxic protein. And then we have gene editing technology, which most of you have probably heard of CRISPR, which is like these little molecular scissors that can come in and cut out specific mutations. And then we use the body's natural repair machinery to try to fix the mutations. So the point here is that based on what your molecular change is and how it impacts your protein structure and function, that really determines what type of therapeutic strategy you might have. Most of these rare diseases that we think of, the molecular change results in a loss of function. So the protein is missing or shortened or degraded. There's not enough of it. So a lot of our therapeutic strategies that we're going to focus on are really this gene addition, modulation, and correction. Some people ask, why can't we just give back the protein? Like, why don't we just go to the end of this process and just give the protein that's missing? And really the reason is twofold. One is that we have this blood-brain barrier that protects. It's a very important job to protect the brain because we want to keep out things like bacteria and fungi and viruses from getting into our vulnerable brain, and it could have really detrimental impact. So we have this special barrier called the blood-brain barrier, but this blood-brain barrier makes it so large proteins can't just float from systemic into the brain. So that makes a therapeutic challenge. If you wanted to give a protein back to the brain, you would have to have, and this has been done like in enzyme replacement therapy, a little like a myoreservoir in the brain, and the protein would be administered directly to the brain to bypass the blood-brain barrier. However, the added challenge is that proteins have something called a half-life. So proteins are being constantly made and broken down and made and broken down and made and broken down. It's not like you just make a protein and it stays there. Some proteins are in your body for just minutes, some for hours, some for days, and some for weeks. And depending on the protein, depends on how often you would have to re-administer it. But most protein type therapies have to be given every couple of days or weeks. So even if you decided you wanted to give the protein and pass the blood-brain barrier, it would be a very regular, repeated administration, which obviously comes with complications to your daily life. And also, every time you access the brain, you have a risk of infection. So just giving back protein directly to the brain or to the body as a whole presents with challenges. That's why a lot of therapies are really targeting the underlying genetic root cause of the disease. And many of these therapies can be one-time treatment or much less frequent than a protein therapy. So one of the most common types of genetic therapies is when we do what's called gene replacement or gene supplementation. So you have your genetic mutation that results in a loss of function of a disease. Most of the time, we know what the one gene is that causes a disease. These are called monogenetic diseases. And what we are trying to do is replace that one gene, and we need a delivery tool. So I'll show more about this, but a lot of times we use viruses, and the viruses get inside your cell, and then they deposit the functional, healthy copy of the gene, and then you start to make that functional protein. So one of the most common viruses used today in gene therapy is called adeno-associated virus, or AAV for short. This AAV, it exists out in nature, and some people have been exposed to it, but it doesn't even make us sick. It doesn't cause the common cold. It doesn't cause the flu. So it's called non-pathogenic. It's a very simple virus. It has these two inverted terminal repeats that look like little T's on the end of this diagram. And then it only has two genes inside it, rep for replication and cap for capsid. So it's a very simple virus, which makes it kind of ideal for using it as a vector for a gene therapy. When we use it for a gene therapy, we gut the virus, we take out the rep and cap genes, and we put in the gene that we're trying to replace for any given disease. So we'll call this the gene of interest. And I know for your indication, there's multiple different genes that cause your family of disease. So the gene you would need to put in would be different for each of the disease-causing genes. And then you have this very important piece called the promoter. The promoter is like the driver or the pilot of the gene, and it determines what cell types the gene is turned on in and what level. Some promoters can be strong and some can be weak. Some can just hit neurons, or some can just hit glial cells, or some can hit all cell types, which is called ubiquitous. AAV has many advantages. It's kind of why it's the gold standard, the most common vector. And one of those advantages I already mentioned is that it's non-pathogenic, so it doesn't make us sick. And another one is that it doesn't integrate into your DNA. Some viruses, like HIV type viruses, they actually integrate into your DNA. And while this is good for stability reasons, meaning that if a cell divides, the virus goes with it, it's not good if the virus accidentally gets into a wrong gene. And some historic cases of this happening with integrating viruses is that they got into cancer-causing genes and then turned on cancer-causing genes, and a side effect of gene therapy was actually getting cancer. So now we have these safer viruses that don't integrate, so they can't accidentally cause cancer, or at least at much, much lower rates than other virus types. We can target these vectors, like I mentioned, by changing out this promoter. They're small. Remember how small this virus is. So some of them can cross that blood-brain barrier, and it's relatively easy to manufacture. One of the disadvantages, and one of the main disadvantages of AAV, is that because it's small, it can only hold about 4,500 of those letters, those base pairs. So some genes are really big. Some genes are like over 8,000 letters. And so for those really big genes, they don't fit inside of this virus. So I'll talk in a minute about some different strategies for those genes. The other disadvantage of AAV is the durability, because remember, since it doesn't integrate, your cell divides and then it will lose a copy of it. And one of the other disadvantages is that if you have been exposed to the virus and you have what's called antibodies, and those neutralizing antibodies can shut down the virus if you receive it as a therapy. It's kind of the same principle as once you get a vaccine that helps your body fight that virus if you see it. So patients that have high antibodies already existing to AAV are usually not eligible to receive AAV as a therapy. With AAV, there's different delivery modalities, different routes that you can give it. And one of those is intravenous, and this is just like giving an injection. And this can systemically treat disease. So if you have a disease that's all over the body, then you might require an intravenous administration. And it's minimally invasive. It's just kind of like giving blood. You don't need any anesthesia. It's a very minimal invasive procedure. However, as I mentioned, you can't have those antibodies circulating in your blood, and the capsid would need to be able to efficiently cross the blood-brain barrier to get into the brain if you need some of that virus to get into the brain. Another disadvantage of giving it intravenous is that a lot of it will go to off-target organs, and namely the liver. Your liver filters things out of your blood, so all of this, you give the virus in the blood, the liver can kind of suck up a lot of it, and that can cause toxicity to the liver if the liver has gets exposed to too much of the virus and can't handle it. Another route is direct organ administration, so you could directly deliver a gene therapy to the brain. This minimizes that off target, so you wouldn't get much to the liver, and you can target very specific regions of the brain, and you don't need to worry about that blood-brain barrier because you're going directly into the brain, and you don't need as much of the virus because you need smaller doses. However, this is very invasive. You're requiring burr holes into the brain, and it doesn't spread far from the injection site. So depending on what area of the brain you would need to target, it could require a lot of injections. So for a large brain, this really isn't feasible for broad distribution, but if there's a very targeted part of the brain for diseases like Parkinson's or AADC, then this can be a viable approach because you know exactly where you need to hit your gene therapy. And then there's kind of an intermediate route, especially for neurologic indications, where we directly target the cerebrospinal fluid, or CSF for short. The CSF bathes the brain and spinal cord, and so it allows us to target the central nervous system and decrease how much is in our peripheral organs like our liver and our heart. And you don't have to give as much as if you give it in the blood. It's minimally invasive. You have to be at least sedated, and it can be given 3 ways: into the ventricles, which is called intracerebroventricular, at the cisterna magna, which is the base of the skull, or lumbar intrathecal, which is like a spinal tap at the base of the spinal column. So this is moderately invasive, but really helps to target more broadly the central and even peripheral nervous system. So these are the ways that we could administer the gene therapy. So this is just kind of a little video showing, what we really do in the lab is try to decide on the AAV serotype, the capsid. There's a few different flavors of the AAV. One of them is AAV9, which is the most common for brain diseases because it's already approved for spinal muscular atrophy. Then we need to decide what we're going to put inside our capsid, which is the gene of interest for your disease and what promoter. Then you inject the virus into the body. Again, this can be done systemically or directly into the CSF. The virus, then, if it's given to the blood, can get out of the blood and across the blood-brain barrier or out of the CSF, and it gets into your cells of interest in the brain. The virus itself is rapidly degraded, but it deposits that gene of interest in the cell. And then the cell with that functional gene starts to then produce the protein that's missing. So the cell starts to become a little factory. There's different types of proteins. Some are enzymes that can be secreted. Some are transmembrane. Some are transporters. So based on the role of your protein, it may or may not be able to get out of that one cell. But the virus can get into other cells. But what we do know is in some diseases that are neurodegenerative, they get worse with age or as disease progresses, these cells can start to get unhealthy and then die off because of the disease process. And if that happens, we can't bring those cells back. Once those cells are gone, they're gone. And so then we no longer have those cells for the virus to get into to serve as factories. That's why we know we need to really deliver gene therapy when as early as possible in diseases that are progressive. So one thing I did want to touch on is that sometimes genes are too big to fit inside AAV. And my understanding is that this is the case for one of the genes in which your foundation is focused on, POLR3A, where you have this gene and because there's so much important information in it, the coding sequence is quite large. And remember, we need the genetic material to be less than 4,500 base pairs in order to fit inside the packaging capacity of AAV. So the POLR3A coding sequence is just over 4,000 base pairs, which makes it difficult to fit inside AAV because remember, you have all this other genetic material, this promoter, which drives the expression, and then you have your it's called your 3' untranslated region where you have some regulatory elements, which really helps the protein figure out what to do once it's made. So those are critical other pieces of information. So if the gene itself is taking almost all the space, then it's difficult to fit these inside of one AAV. So some options in this case are you can make, in some cases it's been successful, you can make what's called a miniaturized gene. You might have heard of microdystrophin, which is the gene that is used in the gene therapy for Duchenne muscular dystrophy. Dystrophin is one of the largest genes and proteins in our body. So in order to make a gene therapy for Duchenne muscular dystrophy, they made a microdystrophin. But why that was feasible for dystrophin is there's a big region of dystrophin that's what's called a repeated domain. It repeats over and over. So they were able to just take out some of those repeated areas and not lose the function of the protein. But with genes like POLR3A, it really looks like almost all of it is important in encoding for part of the protein. So for some genes, it's just not possible to miniaturize it because if you cut out part of it, it would lose its important job. So for some of those that you can't miniaturize, another strategy is to split the gene in half and use a 2-vector system. So you would take one AAV and a second AAV and put the C terminus and the N terminus, so half of the coding sequence, and then hope that once the viruses both get into a cell, that it recombines and makes the full-length functional protein. This tends to be less efficient than just using a single virus system because with a single virus, you just need to know that each cell is going to get that one virus. But with a 2-virus system, each cell will need to get both of the viruses in it in order for the sequences to recombine and make the full-length functional protein. So these are just some strategies for when the coding sequence is too big to fit inside of AAV. There are currently about 7 approved AAVs, AAV-mediated gene therapies. The first one was for an inherited eye disease. So it was just directly injecting the AAV to the eye. And then since that one, it's expanded. And the first pediatric gene therapy was Zolgensma, which is the gene therapy for spinal muscular atrophy. That was developed here at Nationwide Children's Hospital, right here in our Center for Gene Therapy, and the clinical trial was done here. And then expanded to other sites, and now it's FDA approved as of 2019. And over 5,000 babies have received this gene therapy, which has given a lot of safety data, and particularly for one of the capsids called AAV9, because that's what they are using. We also have an approved gene therapy for Duchenne muscular dystrophy that was also developed here at Nationwide Children's Hospital. So as we have more approved gene therapies, the FDA is getting a little bit more comfortable with kind of this developmental pipeline and the safety of these. So we hope that in years to come, we'll continue to see more approved gene therapies. Okay, I'm going to move now from AAV gene replacement and talk about another therapeutic strategy that I mentioned in the beginning. And this is called antisense oligonucleotides, or ASOs for short. ASOs are these short oligonucleotides. They're usually about 20 to 22 base pairs, and they can be used for different functions. You could use them to bind to your RNA to stop protein synthesis, or you can use it to try to alter protein synthesis. I mentioned those splice site junctions before because that's really the easiest way to use an ASO in a loss of function disease. Not the only way, but the easiest way. Because if you just have a misspelling in the middle of your coding sequence in your exon, it's very hard to just put a Band-Aid over a misspelling and change anything. That's why it's usually in these splice site defects where ASOs can be most easily applied. And that was the case for the FDA approved ASO for spinal muscular atrophy. So there's a number of patients that have right in this intron. Remember, introns are usually cut out. They have a defect in this intron between exon 7 and 8. And this caused alternative splicing. So the protein didn't end up getting put together correctly. But if you use this ASO as a little Band-Aid, and then the molecular scissors that were cutting inappropriately now don't see that mistake. And so it allowed for the inclusion of the correct exon, and the protein got put together the way that it should. So this was first tested in a mouse model of spinal muscular atrophy, and they were able to show in the mouse model that it improved the central nervous system, the brain disease, and then went to clinical trial. And now this is an FDA approved drug, and over 14,000 patients with SMA have received this drug. It's under the name of Spinraza. This was a more common mutation that causes SMA, which is a rare disease, but one of the most common rare diseases. And that's why 14,000 people have received it. However, ASOs have really kind of taken on this life of these attempts to make individualized therapies. And so it was the first individualized gene therapy was an ASO, and it was for this patient named Mila who had Batten disease. And so what they were able to do was make the first customized, so really truly personalized medicine. They were able to develop this ASO for this one mutation for this one patient, because nobody else had that same mutation with Batten disease. One of the advantages of ASO is that the drug development pathway not always can be quicker than a gene replacement therapy. Because with ASOs, they don't require making a mouse model of that disease. And the reason is because the splice site defects, they're usually not conserved between a human and a mouse. So the same mutation in a mouse won't cause the disease that it causes in a human. So what they did in the case of Mila is when she got identified with her mutation that causes Batten disease, they made a cell line. So they took cells from Mila, established a cell line, and identified her splice site defect. That's very specific type of mutation that causes improper cutting of the introns and exons. They then screened some different ASOs and identified a hit. They validated the function of the ASO and then tested it for toxicology in rats. So these are just healthy rats where you make sure the ASO is safe and not toxic. And then they got permission from the FDA to start the first patient dosing. Um, Mila was pretty far advanced in her disease at this time, and while it might have attenuated some of her symptoms and bought her some more time, unfortunately, Mila did still pass away from Batten disease. Um, a similar approach was tried in two other patients where they again made an individualized therapy. Again, they're naming them after the patient. So this one was named after a patient named Valeria. And unfortunately, in these next two cases, the ASO caused some side effects, which was when fluid builds up in the brain. And so these two children ended up having a side effect called hydrocephalus. One good thing about ASOs is you can stop the administration. They have to be given routinely about every 3 months because it's not a one-time treatment. It's not lasting forever. And one of the children, they were able to stop it and the hydrocephalus resolved, but then the disease also came back when the ASO was stopped. And the other patient, unfortunately, the patient passed away from the complication of the treatment. So ASOs, they they have great promise, but there's also they can have these side effects that people need to be aware of when developing the ASO. There's a lot of initiatives now surrounding individualized therapy. There's a foundation called In Lorem that's helping to try to develop ASO technology for nano rare patients. So diseases where there's really just very, very, very few patients with it. And there's also another initiative called the N of One Collaborative, which is trying to help kind of streamline the process of ASOs. So the development of ASOs has really kind of taken off in the last 10 years. And unlike gene therapy, where you're putting in the whole gene, ASOs can really be an individualized therapy or personalized medicine, some people call it, where you're targeting those very specific splice site defects. Okay, I'm going to move now to my third and final type of genetic therapy, and this is called gene editing. And after I get through gene editing, I'm going to give some real world examples of some things in my lab, and then we can open it up to some discussion. So gene editing is, again, you're targeting specific mutations, and it's using these molecular scissors that can come in and cut out a mutation. And then what we use is our body's repair machinery to come in and then lay the correct sequence where you cut out the mistake. And when you use that repair machinery, you now have kind of a template and then the correction of the sequence where you used to have a mutation. And so this is very kind of like the ASO, it's mutation specific. So you would need to make a new gene therapy for each of the different mutations that you're trying to correct. Gene editing can be given two ways. One we call ex vivo gene editing. There's also ex vivo gene therapy. You might have heard of that as well. When you're saying that something is ex vivo, what that means is that you're removing the cells from the patient's body. So you take the cells out and then you do whatever your gene editing process is. It could be a gene therapy or a gene editing. Right now, we're focusing on gene editing. So you take the cells out, and then in the laboratory, you put the CRISPR editing into the cells, and then you transplant those cells back into the body. This is really being used for types of blood disorders, so disorders that are dysregulated in the blood, and it's easier to get those blood cells out, correct them, and then put them back. The other is called in vivo gene editing, and this is when you just give the gene editing directly to the body. And really, the easiest organ to target with this is the liver. And the reason is because the liver, all of your blood goes through the liver. So the liver kind of likes to suck up different products, whether they are a virus or in this case, it's a lipid nanoparticle. The gene editing machinery is kind of big. We can't fit it inside one AAV. So we can put it inside of a lipid nanoparticle and then give these lipid nanoparticles just by an IV injection. They will naturally circulate through the liver. And then the gene editing machinery can try to correct liver diseases. You might have heard of that baby KJ at CHOP last year that had a metabolic disease, and they gene edited his liver. And this is how they did. They used lipid nanoparticles and the gene editing machinery. It's much easier to target the liver than it is the brain. And the reason is these lipid nanoparticles cannot get across the blood-brain barrier efficiently. This is an active area of investigation in research labs. If we could get these lipid nanoparticles across the brain, then this same idea might be used for brain diseases. However, right now we would need to put those two viruses to carry all of this information and get them into the brain. And you really have to hit a high number of the cells in order to get efficient gene editing. So while gene editing is here right now for blood diseases and liver and metabolic diseases, it's just not quite prime time yet for neurologic diseases. There is an FDA approved CRISPR gene therapy. And again, this is for targeting blood cells to try to boost the production of fetal hemoglobin. So this has been done, and it is showing really great efficacy for diseases like sickle cell, where you need to try to do the treatment of the blood cells. The very first time in vivo gene editing was tried for a neuromuscular disease where we needed to use those two virus systems. This unfortunately resulted in the death of a patient of Duchenne muscular dystrophy. And this was reported in the New England Journal of Medicine a couple of years ago. So while the technology is promising, it's just not quite there for neurological or neuromuscular diseases. And I think we need some more research to make gene editing safe for those indications. However, I do think it will get there. Okay, so for your different therapies that you're trying to develop, there's really going to be a different roadmap from disease characterization to therapy development. So this is kind of the roadmap that I like to think of, is that first you have to understand the disease mechanism. And you can do this sometimes by just in vitro work, cell culture work with patient-derived cell culture models, which can be really valuable. Then in most instances, we need a mouse model to test our therapy. Then you're going to develop your therapy, whether it's a gene therapy or an ASO or a gene editor. And then you need to determine if that therapy is efficacious, if it has any benefit for the disease, and then what dose to give. If that you need to make sure you have a therapeutic dose, but not a toxic dose. So really establishing that dose range. And then safety and tolerability studies to make sure that your gene therapy doesn't cause any harm. So all this needs to be done before you can translate a therapy to the clinic. And depending on which type of therapy you're using, this roadmap is going to look different. Because as I mentioned with the ASOs, they actually don't require you to make a disease model for every ASO, because especially if it's that splice site, if you change that splice site in a mouse, it's not going to make the human version of the disease. So they actually don't require a mouse model for every ASO development. And it's also going to differ for each disease. So depending on what your disease is and where your starting point is. Are you coming into your therapy development where you already have good model systems that are established and well characterized? Then you can kind of start at the therapy development and use the models that are already characterized. If your disease has just been identified or there hasn't been decades of work establishing model systems, then you might be starting at ground one and you need to develop these model systems in order to test your therapy. So depending on where you're starting will depend on kind of where you need to hop in this map of therapy development. I mentioned that I'm part of ASGCT, so I just want to let you know that we have a big patient outreach branch. We make educational materials. So if you want to know more about gene therapy, there's a whole site of ASGCT where you can watch little videos on what is gene therapy or gene editing. And we are having a Gene, oh, this is some of our patient friendly resources, and we're having an empowering patient um, summit happening March 11th and 12th, if it would be of interest to you. And again, that's my contact, and I will stop screen sharing now to take questions.
I just put mine in the chat, but since I here we have a second here. I had two. One is like the split therapies for the AAV. My daughter has pillar 3A, so I'm just wondering,
Okay.
are those already approved and successful that we've done the split variations?
So there's some different kind of technologies for the split iterations. One of them is coming out of Jeff Chamberlain's lab, and they're trying to use what's called split NTNs, and to put that big dystrophin gene that I mentioned together. And another technology is called RNA enjoining, and that's coming out of the Salk Institute and a company called Insmed. So both of those technologies are moving into the clinic. So that the technologies there, it's getting more efficient. Until the last 5 years, it just wasn't really efficient enough to get those two viruses together. But I do think the technology has improved dramatically, and there will be clinical implementation of two vector systems in the near future.
Thank you. And then I think I was following enough to ask this question, but we know our piece our I guess for the promoters, do we already know that like those have working ones that you know for the oligodendrocytes that we know are kind of impacted with the polar 3A or polar 3?
Yes. So sometimes we put in what's called ubiquitous promoters, which would target, all the cell types in the brain supposedly, but we know that AAV, it primarily, if you don't put a cell type specific promoter, it will primarily go into neurons and secondarily astrocytes. So we can hit some oligos, but it's not great. And so you can put in some different promoters. Like one example is myelin basic protein promoter. There's another one called CNP. And those promoters can help drive the expression more into oligodendrocytes, but oligos are not as easy to target as neurons.
Interesting. Thanks.
Mm-hmm.
No, sorry, can you hear me?
Yes, I can.
Yes, hello. So I have a question. I'm not very good in the genetics, but I'm trying to explain it the best way. So when the genes are too big to fit inside of the AAV, and there are the different pools. So the question is regarding this. So basically, whenever they fix the genes, it's gonna be fixed or the maternal or the paternal mutation, or is it fixed both? So in, let's say, in case when both mutation falls on the same pool, those patients sorry, I'm trying to get here the right way. So whenever
so you're talking about like with
mutation...
a homozygous, like you get one variant from mom and you get one variant
Yes.
from dad, right? Yeah.
Yes.
So once you have those two variants so it's called homozygous recessive, requires two different variants from the two different alleles in order to cause the disease. And so what we're doing with gene replacement, we're actually not touching the gene that you already have that's broken in the cells. We're just providing a new functional copy that doesn't have the mutation, and it would have both of those copies that would be healthy. So for gene therapy, replacement therapy, you're just putting in a new healthy copy. So you're not trying to fix the mother or the father's allele. For gene editing, that's a different question, because if you have two alleles and you're trying to gene edit them, if the mutation is the same on both allele, then you could use one gene editing to fix both of those mutations. If you have two different mutations, like one mutation from mom and one mutation from dad, you'd need to use two different gene editing strategies to target the two different mutations. Sometimes with gene editing, fixing just one of the mutations might be enough, because if you can restore the protein level to say like 50%, that might be therapeutic. So you might be able to fix one, or you might require to fix both, depending on how much benefit you get from just fixing one mutation. Because as carriers, as parents that have one, you have one allele that's normal, it's healthy, and you have one allele that has the mutation that, you know, got passed on. Even with one healthy allele, if you're just a carrier, you have no signs of the disease, right? So sometimes just restoring that one healthy copy can be sufficient.
Right. Okay. Thank you.
There's also a question in the chat about whether you know any about successful therapies specifically for polar 3B related to leukodystrophy.
I don't know any specific, but I haven't dug specifically into that. But Valerie, I think you had that like disease specific table that you were working on, so you might be able to answer that better than I can.
I might just, and Renata, I don't know, tell me if I don't answer your question, but no, right now we don't have anything specific for polar 3B. We don't have anything specific for anything. There are things in development. Dr. Bernard's lab has been working on the polar 3B mouse with replacement, because polar 3B will fit into AAV, into the vector. But I don't know where we stand there. But there is nothing in the clinic yet.
And sometimes that having the mouse model and or the patient derived, in vitro systems are where you're starting at. And if you have an investigator that has that mouse and is willing to work on it, that's a really great starting point. But all those other steps of characterizing the mouse, so you know what the disease looks like in the mouse, and then trying the different therapeutic approaches, whether it's gene replacement or ASOs or gene editing, trying those in the mouse, proving that it works, and then doing the dose response study, and then safety and efficacy testing. So it is not impossible, but it's a long road.
Sam, did you want to add anything to that? I know that you're on there, and I don't mean to call you out, but no. Okay, never mind. Any other questions? Sorry.
Hello, I would have one more question if nobody asked. Has one, so basically whenever you mention that from blood samples, whenever is the research on, and they are able to create these little brain particles, so would that be advisable to look up the research centers and offer blood samples from our children?
You're on mute.
Thanks. If you have researchers that are like actively working on your condition, if they have the ability in their lab to, to use those iPSCs and or make those organoids, those donations can be really valuable. So for like the diseases I work on in my lab, that's, if patients come to clinic here, then we can offer if they want to donate a blood sample so that we can make those. But that process of making the stem cells and then definitely making the organoids, it's pretty expensive and time consuming. So the lab has to have, the resources and the capability to make those model systems. But if somebody's willing to make those, then they are a useful tool. And now there are also some what are called CROs, or contract research organizations, that can do that step for the researchers. Like they can help make the iPSCs, obviously for money, but it can be a useful tool.
I believe, and Valerie, correct me if anybody else knows this, that my brain is saying that VV Hein with the University of Amsterdam was doing that piece of it, because I know in the European leukodystrophy calls that we've talked about, she was highlighting a lot of that research. So if that helps, Agnes, I know that was in flight as of like a year or two ago, that we had the models of seeing the oligodendrocytes like not maturing or the precursor cells and modeling those cells out for that.
And I don't know whether she did that on cells with mutations. I forgot, frankly. But Agnes,
Memory of it was specific to polar 3. I can't remember if it was A or B,
yeah.
but it was modeling those specific oligodendrocytes in that life cycle.
Okay.
So yeah.
And then if you have been at Children's Hospital of Philadelphia, they are collecting samples. So that's an opportunity. And I'm sure that, yeah, there's a possibility to do that through Amsterdam and in other states. So yeah.
Yes, yeah, that was my next question, because we are from Europe, so how we are able to send over any samples. And again, previously when I asked the McGill team, Dr. Brenner's team, they just said to organize it, but it's just need to find out what way we can send the samples for over there, because of it's very hard to post it. So yeah, okay, thank you so much.
Yeah. Right, yeah. I know it's hard to get these things across borders.
Yes, it can. The material transfer agreements and then the custom shipping, and if they're still at the blood stage, it can be really time sensitive. So sometimes it's easier if your home institution could at least get the peripheral blood mononuclear cells or get it to a certain stage before you ship it. So you'll have to work with them for that. Yeah.
I'm writing a note to myself that we need to figure that out. Thanks for bringing it up, Agnes, again.
So far, what I found, it's you have to have different custom forms and stuff, and then again, with the time, within 3 days, it has to reach and go through all the everything. So it just seems so crazy to manage it. And whenever we were in Philadelphia, we were trying to send blood samples to McGill, but I think again it's get lost in the system somewhere, so it's just never happened. But I will keep trying somehow to get that done, and not just for us, for whoever would like to. Thank you.
All right. That was really fascinating, Dr. Bradbury. Thank you so much. And thank you,
A
everybody.
whirlwind tour, but I wanted to get a little flavor of that.
That was great.
That was awesome, though. Thank you.
Yeah.
And thank you for everybody that was able to join us live. And the recording will go up on YouTube. A link will get sent out to everybody via email, and then it'll be posted on Facebook as well. So keep an eye out for that tomorrow. And yeah, hope everybody has a wonderful weekend.
All right. Thank you all.
Thank you.
Bye.
Thank you.
Bye-bye.